And welcome to the 11th session of BGA24. Today we have a session on JBrowse2 with Colin Diesh. Please take it away, Colin.
Thanks so much. I was really happy to get the invite. Damon himself came into our developer chat room and invited me, and yeah, I've had really interesting talks with a number of different people from like Darwin Tree of Life project, and it just sounded like a great initiative going on. So Insta sign up. So really happy to see everyone here. I will try to share screen. I have a number of crazy little disclaimers, which is just that like life has been real crazy. And to add to that, I signed up for the super early session for the USA at least. So this talk has taken on a little bit of what I'm going to call musing. And I'm just going to give you some sort of perspectives on the whole sort of like ecosystem and challenges that we face, and where I think that JBrowse2 fits into sort of the bioinformatics universe. So let's see if I can start the slideshow. Okay. Let's want to start. Does anyone know a hotkey to make it go? Yes. There we go. Okay. So a little more focused now. So yes, I'm Colin Diesh, and I work for University of California, Berkeley with a team of other developers. This is a big group project that we came together to make. But these are just kind of my views only, and what you might expect is that this talk will be somewhat freewheeling, and you're welcome to stop and ask questions anytime, but it's also going to probably be somewhat of a short talk. And so you're welcome to also wait till the end, and we can do any type of extended Q&A. And by the end, you'll see, as I mentioned, how I sort of see JBrowse2 in its place in the bioinformatics ecosystem. And I'm happy to share any type of slides and further content. So the preface to this is that I accidentally signed up for the early morning slot, so it's 4 a.m. for me, and I just got done with a road trip because I've been doing hurricane relief in Asheville, North Carolina, and that's where my partner's from. So it's been real crazy. This was a real disaster for them. So I've had like a lot going on, to, challenge my brain to comprehend the scale of all this. And, there's a lot to say there, and I'm not good with the words. I can throw out that if you have the art or money or voice to do so, feel free to contribute.
Do you have, just while we're on that note, obviously it is a very big tragedy. Is there any particular charity that you would want people to donate to if they are able?
I don't I'm really bad at being in tune. One of them that we talked to was Brother Wolf. They're an animal rescue organization, and they got completely flooded, and they managed to get all their animals out, 100 plus, right before. Everyone had such little warning, but they really came through with that little miracle. So I'm really I can shout out them, but I don't know any other particular orgs.
Okay. I will pop their link in the chat if anyone is interested, and I'll have a look and see if there are any other ones. Because I can imagine they need it right now.
Yeah. So that's where I'm at. And I'm happy to go over that more, but I'll do a little segue right into uh the theme of the talk and give myself a little more background, which is that I'm Colin Diesh, and I got into bioinformatics just kind of randomly through a final project in high school, or sorry, in college, undergrad. And after graduating, I didn't really even know what I was going to do, but I felt like I want to stay involved in bioinformatics. So I just sort of saw this link on Twitter one day, and it said, the Gmod Summer School is going to be happening the 19th to the 23rd of July in Durham, North Carolina. So North Carolina connection there. But I said, I'm going to just sort of sign up for this and see where it goes. It was in person back then in the olden days. And it was a really wonderful experience. I got to meet all the developers of various Gmod tools. Um, they did talks on GBrowse, JBrowse, still new in those days, Web Apollo, um, JBrowse2, all the old classic tools you may have used yourself. And all the developers were there. And so I was like, this is very cool. How could I stay involved even more? So I emailed one of them a couple weeks later and I said, do you guys have any job openings? And they said, yes. So I went and worked on the bovine genome database, who were heavy users of JBrowse and Web Apollo as my first job out of college. And since then, I've just been very sticky and in love with this community. And I got hired to do JBrowse2 as a big rewrite and adventure into this project I'm going to tell you about now. It's been a 6-year-long adventure now, and it's been really cool. So what is JBrowse2? It is a web-based genome browser. And so web-based means it is using things like web technology such as JavaScript, HTML, CSS, and it normally is in a web browser type environment, but it can also run on Electron, which is a desktopified version of it. It's also a genome browser. And so that means it's sort of a tool that conceptualizes data in terms of genome coordinates, which is, relative to a reference genome. And as we know, the whole concept of a reference is sort of getting upturned on its head because of pangenomes, but we still really think about things in terms of genome coordinates, and we're adapting to the new world where there are multiple references out there, and we can review that type of thing. So what is sort of unique about JBrowse2? One thing is that it's able to load multiple genomes at a time in sort of a single instance of itself. So JBrowse1, you only browse one species at a time. And if you want to switch genomes, you can do that, but it goes to a whole new page. It doesn't really have the concept of 2 or more genomes in its little brain at the same time. it can also, as a sort of further extension of this multiple genome concept, show the alignment between multiple genomes, or what we colloquially refer to as synteny, though that has a sometimes finer meaning to people. And we're also able to do some unique things to show structural variants. And so we've worked a lot with the standard VCF format and BAM and CRAM alignments. And somewhat uniquely, it can also have this concept of multiple view types being shown in a single window. So you can create sort of a lot of stuff going on. It's not just the genome browser. You can have, like, this circular view and a spreadsheet and all this stuff going on at the same time. And they can kind of be interlinked too. So you can launch a view from another view, or things of that nature. Excuse me. I'm sorry, the colors are really bad on this. But I said, where does JBrowse2 fit into the bioinformatics code ecosystem? So many tools in bioinformatics sort of look like this. You have, your analysis workflow, you do some stuff, and then you kind of get into R and you start doing your data janitoring and make a cool figure with ggplot2. And maybe that's a twisty path, but then you end up with a paper, and it's one of the figures in your paper. And so that's all very good. And I think this workflow drives a lot of science. It's very powerful. It can be very reproducible. It's great. And one thing that's kind of interesting is that JBrowse2 kind of isn't that in some ways. So, it has this whole sort of interlude where you interactively browse around. So it's very similar. You still might run your analysis workflows, but you then load the data into JBrowse, and you can use our sort of CLI tool or hand edit or programmatically generate the config files. And then you interactively browse around, and then maybe you get to your paper. And, one sort of thought bubble that you might come up with is who loaded the data and who is browsing around. These might be two different people, two different user groups. I'll get into that a little bit more. Because I think it is important to note that, when you're doing back into this old classic static workflow, you have, the person who made the figure and the reader of the paper. But with, say, JBrowse in the middle, you have these different classes of users. You have administrators who kind of set up the instance for you. You have end users who are the people who use the thing that they set up. And you also have people like developers who might just make a plugin for all sorts of people to use. And this distinction, I think, comes out very clearly when you have to write documentation, because it's sort of like, how do you, onboard someone into this system? You have to sort of have these sort of different entry points to help explain it to people. And that's complicated. But I think by creating that distinction, you can kind of get at the best way to explain it. Another thing is that even within this type of distinction, there's a lot of different angles, and that includes different skill levels. And maybe there's people that will barely touch any of sort of the advanced customizations, and then there will people that touch all of them. There's varying knowledge of the file formats involved, and respect of the standards in some cases. It's like GFF can be very chaotic itself. You know, the end users, I just want to highlight that desktop is one of these interesting use cases where people can sort of administer it themselves just entirely through point and clicking all the buttons in the interface. And then the only other small thing I wanted to mention on this different types of developer/admins is that sometimes, you know, JBrowse is kind of just a small part of a larger vision, and it just wants to be sort of a small component of their database. So we try to make it so JBrowse can be componentized and just sort of be incorporated as just a little blurb on your web page or something of that nature. So anyways, designing docs for all these people is hard, but we do that, and you can see the distinction on our so-called sidebar. The user guide, the configuration guide, sort of the administrators, and the developer guide. One random thing I'll throw out is that JBrowse2 is quite a complex app, and it has just a lot of different little features and sort of in-app workflows that you can point and click through. It's just very hard to succinctly summarize all those little things, but they add up to a nice whole, you might say, because, you know, just all those integrations can be powerful. Just to give a sense of the scale, that it's probably approximately 200,000 lines of code that we wrote over the last 6 years. And one thing that's interesting is we need to test it a lot. So it has evolved a lot during that time. Not everything stays the same. So we do sort of these best practice integration tests and unit tests, and then just a lot of manual tests just as we use the app. And some people might also know that they will run into a bug, so we test on our users and they give us a bug, and then we go, oh gosh, I'm very sorry, we fix it. And if you're one of those people, I sincerely appreciate the bug report and the patience with all of this. Always feel free to make those and ping me and whatnot. So this was a funny little slide because I just was like, what, who is my audience? I was really happy to see a bunch of familiar names in the audience, but I'm sure there's people I don't know here, and certainly later viewers on YouTube perhaps. But I think that people are from such a diverse area that it's really wonderful to have these talks with users. And we have this thing that we call our office hours, where you can sign up for a one-on-one discussion with us. It's been really productive. We've had about 40 of them over the past year. We started it in 2023. And of course, we have our GitHub and email and such too. And I just want to give the particular shout out to Darwin Tree of Life, and Yumi Sims has come into our chat and made many interesting, thought-provoking questions. Puzzles, you might say. But it's been really great, and I love to see it. So as I just diverge, this part of my talk I didn't plan very well, because I just thought that I dump a bunch of screenshots at everyone, but I thought I'd just start with like, we did try to think a lot about what we could do to help visualize things like structural variants. And so this is a pretty classic thing where you like have your BAM file and your reads are colored different colors according to which orientation the pairs are pointing. So if it's Illumina, they're supposed to be pointing at each other. But if they get flipped, it kind of like means something's wrong is happening in the genome or the alignment that is fooling it, and that could be a sign of a structural variant. And so the reads are colored to indicate this, and I'm really bad. I need to add we have a feature request to add legends. That would be very useful here so that you could see what blue means, green and teal, but basically it's the pairs pointing one or the other way or outwards. And then there's also sort of the coverage track. And one thing that I actually like about JBrowse is that we kind of count how many reads have things like soft clipping at a particular position. So you can get that sense from these little tiny indicators here, and you can be like, well, maybe there's something going on right there. And people who use IGV, I think, are wizards in that they can just intuit from this weird pattern. But we have a different visualization style in JBrowse2 that we kind of invented called the arc view. And it's actually somewhat new and probably underutilized because it's a little bit of an advanced feature, but it just connects the reads using a arc. And using this, you can actually pretty clearly tell that it's an inversion. Excuse me. Said stomach. So what you can tell is that there's an interesting inside out pattern, I call it. The green is on the left side, sort of on the left of this blue clipping marker, and it's on the left of this blue clipping marker. And the other one, same thing, but it's on the right. So what this kind of indicates is that the green should get moved over here and the blue should get moved over here. And if you did that, it would flip the whole DNA sequence around. The arcs should be small. So you just sort of take the big green arcs, move it together, and then you take the blue arcs and then you move it together, and it just would flip it around. So there's other prior art where bioinformatics tools have done this type of visualization. I was inspired, I think, by Savant browser originally when I made it. But yeah, it's just important to keep re-implementing things that people have done before sometimes, just to keep the state of the art alive, because otherwise the tools fall into disrepair and then suddenly they're on the junk heap and everyone has to use an old version of Java to do their bioinformatics workflows. I think many could relate to that. This is a similar thing. We added it recently, but you can see that you don't actually see the individual reads in this view, but in this one you do. And we just added this. It's a special mode of our so-called breakpoint split view. And so you can see it's the same coloring scheme as before, but it tells you which reads are being connected. And in this particular example, it looks quite nice. The short reads, it's connecting the pairs, and the long reads, it's actually connecting the different parts of the split alignment. So you can think of it like if you have a long read in this region, part of it would align in the forward direction, And then the other part would come back here and align in the reverse direction. And if it's long enough, it would align all the way and get split again and then keep going over here. So that makes you generate a similar, in this case, visualization as you would get it with the short reads, but with much more precise boundaries and niceties such as that. And these are workflows that you can open in JBrowse. I might be able to do that interactively if we have time. I mentioned that this was the breakpoint split view, a new feature of it. The classic sort of version of the breakpoint split view was actually showing the joining of two chromosomes. I probably show this screenshot too much, but it's just kind of cool because it shows you the connection between a read that's split a line between, in this case, chromosome 1 and chromosome 5. And that would just be very hard to do with any type of, genome browser that only shows a single chromosome at a time. I know that IGV isn't necessarily limited to that, but it doesn't do this stacked view. This is another sort of recent feature that we added, still probably not widely used by anyone, but I thought I'd mention it to the power users. You can now do a so-called group by workflow if you're really trying to dig into alignments. It's able to sort of, in this case, split the reads by haplotype. And this is a demo data, so it looks kind of silly, but the reads that are, in this case, haplotype 1 are on the top, haplotype 0. It doesn't have the blue snip on haplotype 0. And then the ones that weren't assigned a haplotype on the bottom. So that's a fun new workflow. This was also somewhat recently added, the ability to filter tracks on the fly using a Jexel expression. Sometimes you kind of just want to there's too many features on your gene track and they're all cluttering up everything. So you can actually type into this field a sort of expression like this, say, give me the feature's name and only show the features that equal this gene name. And so once you hit submit, then it will only show that feature. And there's other types of expressions that you could imagine. So give me only features whose score is greater than 400, or if you have a GFF with all different types of features in it, just only show me the gene features. So that can help you, programmatically clean up your views, kind of similar to some data janitoring in R, perhaps. This is just fun for visual effect. A user has been doing a lot of cool work with Hi-C. I imagine people use this more than I think the Hi-C features, because I personally never have used it a lot, but I know that it's important for genome assembly these days, and it's very cool. So we added a couple new color schemes and the ability to toggle the log scale. So those are just sort of incrementally nice features, and certainly if there's other types of things that Hi-C power users would like to see, I would love to hear it. And this slide isn't very clear, but it shows how basically if you were just browsing in a normal say, linear genome view, you click on a read, then you can see in the feature details panel these links that will launch a new view, a breakpoint split view from it. And I envision more types of things like this. Someone mentioned that they would like to have something like this for Synteny views. So I am continuing to brainstorm things like that because I think this sort of plays into the types of integrations that could be created, is sort of like, yeah, dynamically launching out a new panel or re-navigating an existing panel to a new place. Sometimes that's what you want to do. So it's all that, and this is, you know, just more silly stuff from our demo data. It's our multi-wiggle view, so you can just display a bunch of bigwigs all linked to the same y-axis. If you were using something like R, it does stuff like this automatically, but we have to do a little extra legwork to make that work in the genome browser. And this is something that is upcoming. It was actually merged right before I went down to North Carolina, but I am pioneering a new feature that is basically called multi-way Synteny. So if you're a user of the Synteny views, you'll probably know that it's very pairwise. So you can basically use compare two genomes at a time. And that's quite the limitation if you want to do more than that. It's hard to extend that. But we can do, hopefully now with this new feature, multi-way by conceptualizing it as a series of pairwise. And you can kind of see that here where we have grape versus peach, and then we have peach versus cacao. And there's a little bit of a sort of cognitive overhead to having to toggle those things on manually, so it doesn't sort of automatically scale up to, like, you know, 100 different comparisons. You know, you would then have to toggle on 99 different Synteny tracks. I think that this is a good first step, though, what we have created, and we kind of created the primitive, you might think of, you know, just doing this sort of series of pairwise comparisons. And I think that it will be able to perhaps integrate with cooler, you know, more automatic workflows where you're like, just give me the pan genome of 10 different species and then automatically fill in all the comparisons in between. So then it will just pop up instead of manually opening up this versus this versus this, etc. That's just a little forward looking. And that's actually my last slide. I'm really happy to have had everyone here to join, and I can basically yield the floor to any type of questions. And if there's not really much, I can also try to, like, I don't know, maybe do some live demos, but I don't have anything planned really, so we could just call it a early meeting too. I don't imagine I'll fill out 2 hours.
I know JBrowse has quite a substantial plugin community as well, doesn't it?
Right. And I did not even get into that very much, but one of the interesting things that JBrowse has is the concept of plugins where you can create your own. There's sort of a number of different specific things that are commonly extended by plugins. Like you can make a new what we call data adapter for a custom file format that maybe you made, or you can make a new track type for the linear genome view. So just a custom little visualization, or you can make a custom view type. So you can put an entirely sort of custom view panel, replace this entire thing and just have whatever type of HTML content you want. And, that can it be really interesting. It's like do something that we never envisioned JBrowse would do. I think it's quite cool. I recently have had a long effort to integrate a 3D protein viewer into JBrowse. And it is actually able to sort of have this integration where you mouse over the genome and it shows you where that is on the protein, and vice versa. You highlight the protein and it shows you on the genome. And the cool thing is, AlphaFold DB predicted 3D proteins for everything in Unipro, so a lot of stuff. So you can basically automatically do that for anything that they did, or you can load your own. I'm just going deep into this side project I've been in. Or you can load your own protein that you predicted on Colab Fold or whatnot, and it'll work with that as well.
That's very cool.
Yeah.
See, another camera has turned on.
They have a chat. Is there a chat window? Okay.
It's mostly just me. Yeah, go for it, Angela. You don't have to put your hand up.
Okay. Hello, everyone. Thanks, Colin. This is a really great talk, an introduction to JBrowse. I have a few questions. I'm not sure if it's relevant to discuss it now or for a follow-up meeting, but I was wondering about if there are any suggestions, for example, to improve performance in the visualization of highly fragmented genomes. So for example, in some cases, we will have genomes with, hundreds of thousands of contexts. So it could become, a little bit slow to upload the data. That's one question. Another question, if there is any, recommendation of also how to improve loading of BAM files or visualization of BAM files, because for example, I think like that feature to visualize the breakpoints and inversions and stuff, that feature requires to upload BAM files. So I was wondering if there was a recommendation on that front, because also the examples that you show, I noticed that they were in the order of 1,000 base pairs to 10,000 base pairs. But for example, in some of the genomes that I've handled, like we have 10 megabase long inversions. So that means that we need to upload much larger BAM files. So those were a couple of questions, and then if we have time or interest, I have another one regarding like potential, for example, integration, with Apollo, or like some sort of like, my understanding is that it's possible to kind of like link JBrowse with some sort of tool to do genome annotation or help with the process.
Yeah.
Thanks.
These are all very good questions. So the first one is large numbers of contigs. One of the best things that can help with this is to send me a bug report, because sometimes it can be the most unexpected thing that's slowing it down. And it's just something that we overlooked, and it'll be like, oh, we're just, doing a O of n cubed thing when we shouldn't do that. So if you have an example of this, I would love to see it. There was a recent one where we did find a very silly thing that was slowing it down for large numbers of contigs, and we made a fix for that. That was probably only within the last 2 months or so. So yeah, the other thing is running the latest version. Sometimes you get some surprising little bug fixes of that nature where things just get faster. But I will not say that your problem is probably automatically solved. So I would say there's like strategies where, maybe we need to pare down the set of visible regions to the ones that are larger than 1,000 bases. We don't do that by default because
And they got flooded and they
ghost audio. So yeah, we don't do that by default because we don't want to sort of prescribe anything. But creating those types of filters might help certain use cases, and we could make that. There is an issue for that where we want to ask the user to set a cutoff of some nature. The BAM file example is sort of similar, but it also runs up against some more fundamental challenges. So the web browser is a sort of memory constrained environment. And so it doesn't really like you to just like take all the memory on the computer. So we have to be somewhat smart about things when we try to load a bunch of data. And unfortunately, it's just hard. And so there's some strategies like, we could tell the user only load the reads that are so-called badly paired. So that's a BAM flag, F2 or something. And then that actually would reduce the size of the BAM file a whole bunch, and it would give you the reads that are mostly informative for structural variants. A similar thing can be done with long reads. You filter it for reads that are split. And so that is what I do is I filter all reads that have the SA tag. So that's the split alignment tag, or supplementary alignment tag, SA. So those are kind of like, a lot of people won't do that because it just takes effort to, make another BAM file that, it's like, why can't I work with my existing one? It's just hard for JBrowse to work with the entire data set sometime because it has to load all that into memory. And like the filters that I described, they wouldn't help with that because JBrowse would still load the entire thing into memory and only filter it at the visualization stage. So we don't have a concept of like throwing away everything to avoid loading too much memory. So I hope I am making sense with all that. The other thing, this is a little bit subtle, but if you have, say, a 10 megabase inversion, JBrowse can actually load just the data on either side of it. So that's actually kind of shown in this screenshot. There's this line in the middle, and it has the bases, something to something here, and then something to something here. And it is still connecting the reads across that boundary. So that would be a way to lessen the amount of data loaded, because it's actually only loading stuff here, here, and then connecting them. You will lose what's happening in the middle of that entire 10 megabase region. But that is a possible workaround. Another way to do it is to create, I know I'm really just pulling out all the stops here, but you can create sort of compressed representations, which may be lossy. For example, you can just do a bigwig of the coverage. Sometimes that's actually enough to see things like a CNV. That's a structural variant. And you could also conceptually make another type of bigwig that's a derived data type that shows you the amount of, reads that are badly paired at a certain position. That could give you a signal of an inversion. So JBrowse doesn't have any built-in workflows for that type of stuff, but I think those are really important, especially for like large scale structural variants like that, where you want to get the global picture, you want to see everything that's happening in those 10 megabases, but JBrowse is just going to kind of struggle loading all the reads. But if you create a derived format, say it basically data janitoring it into sort of like a new bigwig file, alternate representation, it might help. And finally, regarding the genome annotation, there is a ongoing effort to make Apollo 3 to integrate with JBrowse2. And there's been some good progress on that, but it is still, say, alpha or even pre-alpha. But I'm happy to give some more info on that. Thanks for the question.
We also have a Apollo 3 session later on in the week. Well, next week, I think.
Right, yeah, my coworker Garrett is going to present on Apollo 3. That would be.
And I think somewhat related to that, I think a lot of the files have like an optional index file that you can also upload, don't they? Which
Yes, so yeah, a lot of the indexes are actually pretty much required if they are available, for example. So we kind of need a BAM index and we need a TabX index for file types that are indexed. So they won't necessarily help performance in that way. They'll just be kind of like needed.
I think, is there going to be any more ongoing support for I think Yumi has probably brought this up with you before, actually. Support for text files and spreadsheets in the browser. Because obviously we can import a text file and have it displayed, but as it have it come up as a track.
Okay, so the specific sort of feature request is convert a spreadsheet
like CSV
to a track.
Yeah.
Yeah, that is a really good idea, and it's one that I've been bandying around. So I don't know. So you guys are one of the users of Spreadsheet View, am I correct? Yeah. So I've been trying to make it's sort of been a slow burner, but a refactor of the Spreadsheet View, kind of aimed at being able to load bigger files because it slows down at pretty moderately sized files and even crashes. Also, I want it to support loading a track into the spreadsheet. So that's kind of the reverse of what you mentioned. And then what you mentioned, which is making the spreadsheet turn into a track, that would be another one. I haven't done that very much. There's sort of an example with that with the SV inspector where it turns it into a circular view track on the fly, but turning it into a linear one would also be great. So yeah.
Very cool.
I will add that note to my sort of slow burner issue on this. Am I really going to run out of battery? Oh, wait. If there are continued questions, I might want to run and get my charger.
I think there's one is well, it seems like a bug report on not being able to install on Windows because the exe can't be found, or in Linux it's a C++ error, isn't it? I came across this today for something else.
This is really interesting. I would definitely recommend reporting that via email, or GitHub even, but email's fine. You know, if you want, my personal email is colin.diesh@gmail.com. Welcome to the email there, but GitHub as well. Yeah, and then there's the security on Windows, which is very strict and annoying, and sometimes you can bypass that if that is the issue.
So I'm just trying to, I've lost the YouTube stream, so I'm just trying to find that and see if anyone.
I saw one from Tias. He said, are there any best practices for hosting a publicly available JBrowse to accompany a publication? And I would say there's not really too many. If you can put it on a web server, you know, there's such classic ones as Apache and Nginx. Those work fine. If you want to play around with sort of fun modern technologies, there's Amazon S3, which doesn't even require hosting a server that is, you know, just sleeping all the time. So S3 is just a static file hosting, and JBrowse can be entirely hosted from there, which is quite cheap. So you don't even pay $5 a month. You can pay like pennies on the month. That's fine. Thanks, Colin. But you don't have to.
Well, I think there's another one from Sam. Is there a demo for the 3D protein plugin? There's a GitHub page, but no demo. I think he's probably disappeared now, hasn't he? Right. In that case, I imagine his laptop has probably died by now. If there aren't any more questions, then please drop them in the Discord, and I'm sure he'll answer them whenever he gets back. Alternatively, there is the JBrowse support page that people can drop tickets into and have chats with the developers on. I don't know, that's thrown me off my groove now. My usual spiel at the end of these videos. Yeah, there's the Discord, there's the YouTube, there's plenty of demos on the website, although I don't think the 3D one is on there, evidently from Sam. And I suppose I'll see you all in the next session. So thank you very much for attending.